Morning. Today we are going to see about cell cycle and cell division. Cell cycle, from one cell, how the whole cycle goes so that it ends up in two cells. That's what we are going to see today. Okay. And next we'll see about the mitosis and meiosis also. So what is cell cycle? Cell cycle is a cell division is an essential process in all living organisms. The cell divides. Both are exactly same as the mother cell. The mode of cell division is fundamentally similar in all organisms. During the process of division of cell, the processes like DNA replication and cell growth must take place in a sequential and coordinated manner to ensure the correct division and formation of progeny cells with intact genome. Cell cycle is an orderly sequence of events or a set of stages by which a cell duplicates its genome synthesis, the other constituents of the cells, and eventually divides into two daughter cells. In terms of cytoplasmic increase, the cell growth is a continuous process while DNA synthesis occurs during a specific stage of cell cycle only. The DNA, that is the chromosome, are further distributed to daughter nuclei by a series of events. Okay, so we are going to slowly see what are those events. A typical eukaryotic cell, that is a human cell, our cell, divides once in every 24 hours. This duration of cell cycle can vary from organization to other organisms and also from one cell type to another. For example, in a yeast cell progresses through the cell cycle and divides in only about 90 minutes. Within 90 minutes, you can see a new cell develops. Interphase. It is the period between the end of one cell division to beginning of the cell division. That is between two successive M phase. M phase. During this phase, cell prepares itself for both cell growth and DNA replication in an orderly manner. So, it is also known as preparation phase. It is also called as preparation phase. It lasts for about 90 to 96%, more than 95% of the total duration of cell cycle. Okay? In a cell cycle of a human cell, only about 1 hour is taken by the dividing phase out of 24 hours duration of one cell cycle. Interphase is further divided into following 3 sub-stages. Okay? And how they are divided? It is based on the various synthetic activities. First is the G1. That is gap 1 phase. G1 or gap 1 phase. It corresponds to the duration between the mitosis M phase and initiation of replication DNA. The cell becomes metabolically very active during this period. Okay? It grows continuously and prepares itself for DNA replication thus synthesizing enzymes and proteins needed for this process. Okay. Now look at the S phase. S phase is a synthesis phase. It is known to be the phase in which actual synthesis or replication of the DNA takes place. Replication from 1, it becomes into 2. The overall amount of DNA doubles per cycle, but no increase in chromosome number takes place. during this phase. So there is no increase in the number, it only replicates. If the initial amount of DNA is 2C, it will become 4C. In case of animal cell, during S phase, DNA replications begins inside the nucleus while the duplication of centrioles takes place in the cytoplasm. Now look at the G2, gap 2 phase. This phase is also called post-synthetic or pre-mitotic phase. During this stage, the synthesis of DNA stops and proteins required for mitosis are being synthesized while the growth of cell continues. It prepares the cell to undergo divisions. Next is the M phase. Following the interphase, the cell enters the M phase or the mitotic phase. It is also called as mitotic phase, that is the M phase. Next, in this type of division, the chromosomes replicate themselves and get equally distributed into daughter nuclei. Example, the chromosome number in the parental and progeny cell that is diploid, becomes the same. It is known as equational division. Mitosis is also known as somatic cell. It's also known as somatic cell division because it always occurs in somatic cells. Mitotic cell division is seen in the diploid somatic cells in animals, whereas in plants, mitotic division is seen in both haploid and diploid cells. It is known to be the phase of actual cell division, which starts with the division of nucleus followed by the separation of daughter chromosomes, that is karyokinesis, and terminates with the cytoplasmic division. That is cytokinesis. Next slide. It is divided into four main substages. We call it as a prophase, metaphase, anaphase, and telophase. Okay. Now look at each phases. Prophase. This phase is known for initiation of condensation of the chromosomal material, which during the process of chromatin condensation becomes untangled, and finally the centrioles begins to move towards the opposite pole of animal cell. Opposite poles of the animal cells. We can categorize prophase as early prophase. During this phase, so what is early prophase? During this phase, condensation of chromosomal materials takes place in order to form a compact mitotic chromosomes composed of two chromatids, which are attached together at centromeres. The most conspicuous change that take place during prophase is the formation of mitotic spindle. Remember that, the mitotic spindle. Okay. The initiation of mitotic spindle assembles the microtubules and the proteinous compounds of cell cytoplasm, helps in the completion of the process. The mitotic spindle is formed between the two pairs of centrioles that migrate towards the opposite poles of the cells. Now look at the late phase. So what is the late phase? At the end of the prophase, late prophase, at the end of the prophase, during late prophase, the nucleolus disintegrates gradually and the nuclear envelope disappears. Nuclear envelope disappears. This disappearance marks the end of prophase. Okay? Disappearance of the nuclear membrane marks the end of prophase. Next is metaphase. It is a phase that starts after the disintegration of the nuclear envelope in the late prophase. Okay? The chromosomes spread out through the cytoplasm of the cells and are seen to be shortest and thickest. They are seen to be shortest and thickest. The chromosomes can be easily observed under the microscope. You can see it under the microscope. Each chromosome at this stage is made up of two longitudinal threads and are held together by centromeres. Centromeres in the center. At the surface of each centromere, disc-shaped structure called kinetochores are present. Kinetochores are present, which help in the attachment of spindle fibers to the chromosomes. Attachment of the spindle fibers to the chromosomes. The chromosomes finally arrange themselves at the equator in one equatorial plane known as Metaphase plate. It is called as Metaphase plate. Attachment of the spindle fibers to kinetochores of chromosomes. Number 2, movement of the chromosomes to the spindle equator and it is alignment along the metaphase plate through spindle fibers to both the poles. Okay. Next comes the anaphase. It is known to be the shortest duration, that is about 2 to 3 minutes, and is also very simple stage. At the beginning of this phase, splitting of chromosomes takes place. Splitting of chromosomes takes place. The two daughter chromatids now become the chromosomes of future daughter nuclei. Okay? Chromatids now become the chromosomes of future daughter nuclei and starts migrating towards the opposite pole along the path of their chromosomes, along the path of their chromosome fibers. Okay? During anaphase, what are the things that happens? Number 1, splitting of the centromere and separation of the chromatid. Splitting of the centromere, separation of the chromatids, both occurs. Movement of the chromatids to the opposite poles. 3 is movement of the chromatid to the opposite poles. Okay. Next and the final thing is the telophase. This is considered to be long and complex phase like the prophase. The final stage of mitosis. Okay. At the onset of this stage, the spindle fibers disappears. The spindle fibers disappears. The chromosome decondenses and further lose their individuality after reaching their respective poles. In general terms, the events of prophase occur just in reverse sequence during this phase. Now, the individual chromosomes cannot be seen and chromatin materials gets collected in the form of mass in both opposite poles. Thus, the changes are observed during telophases. And thus, what are the changes that are observed during telophase? Number 1, the chromosomes gradually uncoils and clusters at the opposite spindle poles. Thus, their individual identity as discrete element is lost. Okay, so the individual identity as a discrete element is lost. Nuclear envelope slowly reforms around the chromosomes, nuclear membrane. Okay. Reappearance of nucleolus. Reappearance of nucleolus. Golgi complex and endoplasmic reticulum also takes place. Okay. That finishes telophase. It's commonly known as the division of cytoplasm of the parent cell into two daughter cells after the division of nucleus or karyokinesis. It occurs so that each daughter cell can receive nucleus of its own. Okay. Thus, mitosis is not only the segregation of nucleus into two daughter nuclei, the cell also divides itself into two daughter cells. Cytokinesis in animal cells. Okay, so what is cytokinesis? In animal cells, cytokinesis starts at metaphase. They typically divided by furrowing, by the appearance of the furrow in the plasma membrane. This is also known as cleavage. Due to the contraction and development of microfilaments, a constriction develops which further deepens in a centripetal way known as cell furrow. It is known as cell furrow. The furrow starts deepening gradually during telophase and finally gets joined in the center by dividing the cytoplasm into two. Cytokinase in the plant cell. Cytokinase in plant cell is different from that in the animal cells due to the presence of a solid, rigid and inextensible cell wall on the outer side of the cell. The plant cell cannot undergo cytokinesis. By furrowing method. Therefore, plant cell divides by cell plate method. It is called a cell plate method. The formation of cell plate usually begins during the late anaphase or early telophase. The formation of new wall in plant cells takes place in the center of cell and starts growing outwards towards the opposite sides in order to reach the already existed lateral walls. This new cell wall with the formation of simple precursors grow until it reaches the actual cell walls. Once cell plate has divided the cell into two cells, it will continue to grow and develop into a new cell organelles and also get distributed among two daughter cells. During the cytoplasmic division or cytokinesis. Cyto means cell, kinesis means regeneration. Syncytium cell. In some organism, karyokinesis is not followed by cytokinesis. Therefore, only nuclear division takes place, not whole cell divides. As a result, multinucleated condition arises leading to the formation of syncytium. Example, liquid endosperm in coconut as well as in selenocytic hyphae of lower fungi. Okay. Next, mitosis has following significance. What are the significance of mitosis? Let us understand that. It helps in production of diploid daughter cell with each an identical genetic complement. Each identical genetic complement. Mitosis helps in growth of multicellular organisms. It also helps in maintaining a proper cell size by dividing an overgrown somatic cell. It is helpful in cell repair mechanism. Example, continuous replacement of the cell, like of the upper layer of epidermis, cells of the lining of the gut and blood cells. Cells of the lining of the gut and the blood cells. The growth of multicellular organism is due to mitosis, as it is known that a cell cannot grow in size beyond a certain limit without disturbing the ratio between the nucleus and the cytoplasm. Okay? After reaching the particular size, the cell divides in order to restore the nucleocytoplasmic ratio. Therefore, the growth of the cells takes place by increase in the number of cells, rather than the increase in the size of the cell. It's increase in the numbers. Okay? It is also helpful in producing new cells for healing wounds and for regeneration. Mitosis plays an important role in continuous growth of the plant throughout their life by meristematic tissue, tissues like the apical and lateral cambium. Okay. Now we have finished the study of mitosis. We are moving on to meiosis. Meiosis is the phenomenon on which occurs in any life cycle that involves the process of sexual reproduction. The production of offspring by sexual reproduction involves the fusion of two gametes. These gametes are formed in male and female reproductive organ. So, these gametes are meiotic cell division. They are all formed by meiotic cell division. Thus, meiosis is known to be specialized form of cell division which reduces the chromosome number in such a way that each daughter nuclei receives only one set of each kind of chromosomes. That is maternal and paternal. It results in production of haploid daughter cells. Haploid, just the half. Haploid daughter cells. In meiosis, the nucleus divides twice, but the replication of chromosomes takes place only once. Got my point? Interphase of Meiosis 1. Thus, it is also known as reductional division. The essential feature of meiosis are: number 1, meiosis undergoes 2 successive cycles of nuclear and cytoplasm division. That is meiosis 1 and 2. Meiosis 1 and 2 occurs one after another with a very short or no interphase at all. After the replication of parental chromosome, initiation of meiosis 1 take place in order to produce identical sister chromatids at S phase. Meiosis takes place about days to get completed instead of hours or minutes that are needed for mitosis. It takes days to be completed. Okay. Pairing of homologous chromosome and recombination takes place during meiosis. Okay. Finally, after the second meiotic division, 4 haploid cells are formed. 4 haploid cells are formed. Okay. The homologous chromosomes of each pair separates from each other and reaches separate daughter cells, which thereby reduces the number of chromosomes from diploid to haploid. Reduces the number from diploid to haploid, from 2n to n. Thus, it is known as heterotypic division. Heterotypic division. It is further divided into 4 phases. That is, prophase 1, metaphase 1, anaphase 1, telophase 1. Okay. Now, prophase 1. It is considered to be the most complicated, prolonged phase as compared to similar stages in metaphase. This phase is further subdivided into 5 subphases on the basis of chromosomal behavior. Remember the name. Leptotene, zycotene, pachytene, diplotene, and diakinase. So what is leptotene? It is known to be a very 5 stage of meiotic division following the interphase. Okay. Chromosomes becomes gradually visible under light microscope. Centriole starts moving towards opposite end or poles, and each centriole develops develops the race. Each chromosome is attached to the nuclear envelope through the attachment plate at both of its ends. Zygote. This is the next stage that takes place after the completion of previous ones. After the completion of the previous one. This is also a short-lived stage like leptotene. Okay. What are the changes that are seen in the phase? Okay. So, homologous chromosome pairs up. This pairing is done in such a way that genes of same character present on the 2 chromosomes lie exactly opposite to each other. This process of association is known as synapses. It is revealed from the electron microscopic studies that the formation of synaptoidal complex takes place by a pair of homologous chromosomes that show synapses. The complex so formed on account of synapses forms a bivalent or a tetrad. Okay. Next is the pachytene, the stage which immediately follows zygotene, where the pair of chromosomes become twisted spirally around each other and cannot be distinguished separately. This stage is comparatively long-lived as compared to the previous 2 stages. Got it? Following changes are seen during this particular stage. What are the following changes, right? Bivalent chromosome clearly seen as tetrads. Bivalent chromosomes are clearly seen as tetrads. In this stage, sometimes exchange of gene or crossing over between the two non-sister chromatids of homologous chromosomes occur at the points called recombination nodules, which appear at intervals on the synaptimal complex. Diplotin. It is the stage of longest duration of all. So what are the changes that happens? Let us see. In this synaptimal complex appears to get dissolved while the chromatids of each tetrads remains clearly visible. So chromatids of each tetrad clearly visible. Recombined homologous chromosomes of the bivalent get separated and form chiasmata, that is egg-shaped structure. The chiasmata, egg-shaped structure. Chiasmata formation is necessary for the separation of homologous chromosome which have undergone the process of crossing over. Diakinesis. This is known to be final stage of meiotic prophase 1, also known as terminalization due to shifting of chiasmata towards the end of the chromosomes. Okay, chromosomes become fully condensed. Nucleolus degenerates. Breakdown of nuclear envelope into vesicles occurs. Formation of meiotic spindle in order to prepare the homologous chromosomes for separation also occurs. Diaconas phase represents the transition from prophase to metaphase of Meiosis 1. Metaphase 1, it is the stage followed by prophase. So what are the changes that happens here? The bivalent during this phase arrange themselves on two parallel equatorial plates. The centromeres projects little bit towards periphery. Since there are two centromeres in each bivalent. Thus, each centromere is joined by chromosomal fibers. The fibers of the homologous chromosomes are always in the opposite directions. Anaphase 1. Anaphase 1 is the next phase after metaphase 1, in which homologous chromosomes break their connections with each other and get separated. This process of separation of homologous chromosome is known as disjunction. It is called as disjunction. The separated chromosomes are univalent and are also called dyads. Next is the Telophase 1. This is the last stage of meiosis. Meiosis 1, in which the chromatids are at each pole of spindle usually remains uncoiled and get elongated. Look at the changes that happens here. Homologous chromosomes reach at their respective poles. Reappearance of nuclear membrane and nucleolus takes place. Nucleolus takes place. Cytokinesis, it is the stage during which the cytoplasm and other organelles divided into two equal halves of cells. Interkinesis. This is a stage between the two meiotic division. Okay, two meiotic division. The Meiosis 1 and 2. It is generally short-lived. During this process, no replication of DNA occurs. It is necessary for bringing true haploidy DNA in daughter cells. It is in fact considered as incipient interphase. It is considered an incipient interphase. Meiosis 2 is known by another term, homotypic division. It is also called as homotypic division. It is often known as equational division. Meiosis 2 also resembles a normal mitotic division in contrast to Meiosis 1 because it distributes chromatids to daughter cells. Prophase 2, this is known to be very short stage out of all. So what are the changes that happens in prophase 2? The centrioles duplicates themselves by separation of two members of the pair. Each chromosome comprising two chromatids. Each chromosome comprising two chromatids becomes visible in the nucleus. These chromosomes further become thick and short in size. Nuclear envelope breaks down and formation of spindle apparatus takes place and nucleoli disappears. Metaphase 2. Okay. This is the next phase, and what are the things that happens here? Let us look at one by one. The chromosomes align at the equator or the metaphase plate. It is called as metaphase plate in the similar way as mitosis, normal mitosis. Chromosomes get attached to the fully formed spindle apparatus and kinetochores of sister chromatids for each chromosomes face the opposite poles and each is attached to kinetochores, microtubules coming from the pole of that side. Anaphase 2. This is the phase followed immediately after metaphase 2. Okay. Look at the changes that happens here. So what are the changes? Number 1, centromere of each chromosome splits that was holding the sister chromatids together. Number 2, the shortening of chromosomal microtubules. Shortening of the chromosomal microtubules takes place. The two chromatids of each chromosome start moving away from each other and finally reach the opposite poles of the spindles. They reach the opposite poles of the spindle. Telophase 2. This is known to be the last stage of second meiotic division and shows changes equally opposite to that of the prophase. 2. Meiosis ends up with the progress of this particular stage, this particular phase. Telophase 2. That is the telophase 2. So it ends with the telophase 2. Formation of nuclear envelope around each set of chromosomes. Formation of the nuclear envelope around each set of chromosomes. Nucleoli reappears due to the synthesis of the ribosomal RNA and ribosomal DNA. Cytokinesis. This occurs after every nuclear division. It includes separation of cytoplasm and organelles in two halves of the cells, into two halves of the cells. The two daughter cells formed have half the number of chromosomes and the amount of nuclear DNA. Both the cells undergo divisions and give rise to 4 cells. These haploid cells are arranged tetrahedrally and collectively called tetrahedral or tetrads. We call it as tetrads. So what are the significance of meiosis 2? It maintains the same chromosome number in sexually reproducing organism from a diploid cell to a haploid gametes are produced. Diploid cell to haploid gametes are produced, which in turn fuse to form a diploid cell. Very interesting. Haploid gametes are formed due to the reduction of chromosomes to its half. It restricts the multiplication of chromosome number and maintains the stability of the species. Maternal and paternal genes get exchanged during crossing over. It results in variation among the offspring. All the 4 chromatids of a homologous pair of chromosomes segregate and go over separately to 4 different daughter cells. This leads to variation in the daughter cell genetically. Paternal and maternal chromosome assort independently. This causes reshuffling of chromosomes and changes traits controlled by them. Such an important phenomenon that has maintained all the living creatures throughout the world. So if you understand mitosis, meiosis 1 and meiosis 2, we will enjoy this world, a beautiful world. Why? We are all different in this world. I am different from my sister. My sister is different from her daughters. And just that goes on. Beautiful, isn't it? Let's work together to make this world much more beautiful. Thank you.